Welcome to Serenity and Fire, the podcast where wellness meets grit. I'm your host, Krista Guidenti, founder of Panacea Luxury Spa Boutique. Join me as we dive into the intriguing world of biohacking, clean living, cutting-edge spa trends, and the hustle, grind, and grit of entrepreneurship. From my personal battles with weight management, infertility, and the 30-year journey to create and launch my dream business, to building a sanctuary for those who have been touched by cancer, I'm here to share real talk, inspire big dreams, and spark a passion for holistic living inside each and every one of you. So let's dive in. Welcome back to Serenity and Fire. Today, I wanted to start by asking you a question. What if the brain could heal itself? Think about that for a minute. Because when we talk about Alzheimer's disease, Parkinson's, dementia, stroke, traumatic brain injuries, or even the cognitive changes that many of us notice as we age, we're talking about something far more consequential than forgetting where you put your keys. We're talking about the health and survival of the cells and the connections that allow you to remember, reason, move, communicate, make decisions, recognize the people you love, and ultimately to remain you. For decades, much of medicine has focused on managing the symptoms that occur when those systems begin to fail. But researchers have also been pursuing a much more ambitious possibility. What if we could protect vulnerable neurons before they're lost? What if we could help injured neurons recover? What if we could preserve, or perhaps even encourage, the connections between brain cells? And could we potentially influence some of the biological processes involved in neurodegenerative diseases before enormous amounts of damage have already occurred? That's where today's peptide conversation begins. So welcome back to part 7 of this crazy and exciting series. Up to this point, we've explored peptides involved in metabolic health, healing and tissue repair, and most recently, the growth hormone system. Today, we're going somewhere completely different, the brain. But before we can possibly evaluate whether a peptide can help heal the brain, we need to understand what healing the brain would actually require. What happens when neurons become damaged? What happens when the connections between them begin disappearing? Can the brain build new connections? What allows it to adapt after injury? And what are researchers actually trying to influence when they talk about things like neuroplasticity, BDNF, NGF, GABA signaling, or synaptogenesis? Once we understand what we're trying to repair, we'll be much better equipped to evaluate whether any of these so-called brain-healing peptides can actually do this job. So today, we're going to build the foundation. We're going to talk about how neurons communicate, what a synapse actually is, what neuroplasticity means, what BDNF and NGF do, why GABA matters, what HGF-CMET signaling has to do with neuronal growth and repair, and perhaps most importantly, what is actually going wrong inside the brain when we're talking about Alzheimer's disease, Parkinson's disease, and the different forms of dementia. And then throughout the next couple of weeks, we'll put Cmax, C-Lank, dihexa, and cerebrolysin through both our Serenity and Fire peptide checklist and my promise versus proof scale. We'll look at cognition, anxiety, stroke and brain injury, Alzheimer's, Parkinson's, dementia, healthy brain optimization, and as always, any legitimate questions surrounding cancer. But today, before we ask whether these peptides work, let's understand what we're asking them to do. And also, before we look at something that someone might inject, spray into their nose, or take in another form, we need to define what we mean when we say something could actually heal the brain. Because I'm sure you would agree that that's an enormous claim. Your brain contains roughly 86 billion neurons, but those neurons don't function independently. They're constantly communicating through an extraordinary network of connections. And one of the most important of these connections is called a synapse. A synapse is essentially the communication point between neurons. One neuron sends a signal, another receives it, and those networks allow information to travel throughout the brain. If this seems a little bit elementary to you, just hang in there with me because the science does get a little complicated, and I just want to make sure that everyone listening can understand what we're talking about as we dive into these peptides. So memory depends on these networks. Learning depends on them. Movement depends on them. Emotion, attention, decision-making, language, they all rely on incredibly complicated networks of neuronal communication. So maintaining brain health isn't simply about keeping individual brain cells alive. We also need to preserve the connections between them. And that introduces a word you're going to hear a lot throughout the rest of this brain healing peptide series. And that word is synaptogenesis. Synapse refers to the connection. Genesis means creation. So synaptogenesis literally means the formation of new synapses. In plain English, building new connections between neurons. And when you hear someone making an extraordinary claim that a compound like dihexa, for example, promotes synaptogenesis, that's what they're talking about. They're not claiming that we're simply giving an old neuron more energy. They're talking about potentially influencing the machinery involved in building neuronal connections. You can immediately understand why scientists interested in Alzheimer's disease might find that fascinating, because Alzheimer's isn't simply a problem of bad memory. Synapses actually become dysfunctional and are lost. Neurons eventually die and brain networks progressively deteriorate. Imagine if we could protect some of those connections or encourage the brain to establish new ones. That would be an entirely different therapeutic strategy than temporarily improving a symptom. But, and we're going to encounter the distinction repeatedly today, demonstrating synaptogenesis in cells or animals is not the same thing as demonstrating that you reversed Alzheimer's disease in a human being. Keep that distinction in the back of your mind because it will become important throughout this series. So now I want you to meet the brain's growth and survival signals. Let's start with BDNF and NGF. BDNF stands for brain-derived neurotrophic factor, and NGF stands for nerve growth factor. The word I really want you to remember is neurotrophic. Think of neurotrophic factors as some of the brain and nervous system support and maintenance signals. They can help neurons survive, develop, adapt, and form or strengthen connections. BDNF is especially involved in neuroplasticity, or the brain's ability to change and adapt in response to experience. Learning something new, your brain has to change. Forming a memory, networks have to adapt. Recovering function after certain forms of brain injury, again, the brain has to reorganize. BDNF participates in that biology. NGF has its own roles, particularly in the growth, maintenance, and survival of certain neurons. So when you hear someone say, Cmax increases BDNF, I don't want you simply thinking, great, BDNF is good, more must be better. I want you asking, where was that demonstrated? In cells, in an animal, in human blood, in the human brain? And most importantly, did increasing that biomarker actually improve the patient's health? There is human Cmax research in post-ischemic stroke rehabilitation reporting increased plasma BDNF alongside better functional recovery measures, for example. That's considerably more interesting than merely demonstrating a molecular change in a petri dish, but it still doesn't establish Cmax as an Alzheimer's treatment. Another system involved in brain biology that will be important for you to understand as we talk about this brain peptide category is GABA. GABA stands for gamma aminobutyric acid. It's one of the brain's major inhibitory neurotransmitters. And inhibitory doesn't mean bad. Think of your nervous system as constantly balancing go signals and slow down signals. GABA is one of the brain's primary ways of saying, let's turn down the volume a little bit. That matters for anxiety, stress response, sleep, neuronal excitability, and the overall balance of brain activity. So when I use the phrase GABAergic signaling today, all that means is communication in the nervous system involving GABA. You don't have to remember the word, just remember GABA helps regulate the brain's brake pedal. That's going to become especially relevant when we get to C-Link because research suggests one of its anti-anxiety mechanisms may involve modulation of GABA receptors. Small human studies have also evaluated C-Link in anxiety disorders, although that is a very different evidence base from proving that it prevents dementia or neurodegeneration. And then there's HGF-CMET, and it's spelled HGF-C-MET. One final pathway, and this one deserves special attention. HGF stands for hepatocyte growth factor. Hepatocyte literally refers to liver cells, which is where this growth factor was first characterized. But HGF does far more than act on the liver. Its receptor is called CMET. When HGF binds to CMET, it activates signaling involved in things like cellular survival, growth, movement, repair, and in the nervous system, processes related to neuronal development and synaptic connectivity. So why do we care about any of that? Because dihexa, which is a peptide, appears to interact with the HGF-CMET system. There's some research that I'm going to share with you that sounds a little bit complicated, but just stick with me. So preclinical research found that dihexa, which is one of the brain-healing peptides, enhanced HGF-dependent c-Met activation and promoted new dendritic spines and synapses in hippocampal models. Blocking HGF c-Met signaling also blocked its procognitive effects in the animal model. You're probably saying, okay, what the heck was all of that? I know it's intense and you may not have any idea what it means, but I'm going to break it down here for you. Remember that HGF c-Met pathway that we just talked about? Think of it as one of the cellular communication systems involved in growth, survival, and repair. Dihexa appears to enhance that signaling. And in laboratory and animal models, researchers saw something else. Neurons began developing more of the tiny structures, or those dendritic spines, that allow them to form connections, or those synapses, with other neurons. In other words, the brain cells appeared to be building more places where they could communicate with one another. That's synaptogenesis. And researchers did something particularly interesting in this study to figure out whether the HGF c-Met pathway was actually responsible for that synaptogenesis. They blocked it. And when they blocked that pathway, the cognitive benefits in the animal model disappeared. This was a very powerful clue that the HGF c-Met signaling that's enhanced by dihexa is the very thing that produced these desired effects. But, and keep this distinction in mind, we're still talking about preclinical research. Helping neurons form connections in laboratory and animal models is fascinating, but it's not the same thing as demonstrating that dihexa can rebuild lost neural connections or reverse Alzheimer's disease in a human being. On paper, it sounds extraordinary. A compound that might help neurons form new connections, who wouldn't be excited about that? And you can immediately see why people interested in Alzheimer's and dementia also became excited about it. But HGF c-Met is also where our cancer conversation gets much more serious, because cellular growth, survival, and movement are wonderful things when you're repairing healthy tissue. But those same biological capabilities can become dangerous when they're exploited by a malignant cell. So with dihexa, we aren't merely going to ask, could this help the brain? We're also going to ask, what else might we be stimulating elsewhere? And do we have enough human safety data to know? Those questions may ultimately matter just as much as the promise with this compound. So to summarize all of this brain biology, synaptogenesis means building neuronal connections. BDNF and NGF are neurotrophic factors involved in supporting neuronal health and plasticity. GABAergic signaling refers to communication involving one of the brain's primary inhibitory neurotransmitters. And HGF c-Met is a powerful growth and survival signaling system with intriguing implications for both neurodegeneration and cancer biology. Okay, so now that we have all of the brain vocabulary sorted out, we can actually understand the science I'm about to share with you. But there's another piece of this conversation that we need to understand before we really start diving into the individual peptides. I keep mentioning Alzheimer's, Parkinson's, dementia, and neurodegeneration. But one thing I want to clarify is that these aren't interchangeable terms. And if we're going to ask whether a peptide could someday help protect or even repair the brain, we first need to understand what exactly is going wrong in the brain that we're trying to fix. So let's start with one really important distinction, and that's that dementia is not one specific disease. Dementia is an umbrella term, and it describes a decline in memory, thinking, reasoning, language, or other cognitive abilities that becomes significant enough to interfere with someone's ability to function independently in everyday life. Several different diseases can actually cause dementia, and those diseases don't all damage the brain in the same way, which means there probably isn't going to be one universal solution to dementia. So let's look at the 4 major conditions we're going to reference throughout these next few episodes. Let's start with one that most people immediately associate with dementia, and that's Alzheimer's disease. Alzheimer's is the most common cause of dementia. And although we still don't completely understand every step that causes the disease to develop and progress, we know several things are happening inside of the brain with Alzheimer's. You probably heard of amyloid plaques. Amyloid beta is a protein fragment that can accumulate between neurons and form abnormal deposits called plaques. You've probably also heard of tau tangles. Tau is normally a protein inside neurons that helps stabilize part of the cell's internal transport system. But in Alzheimer's disease, tau can become abnormal and accumulate into tangled structures inside neurons. So you've essentially got abnormal protein accumulation happening both around and inside the brain cells. But here's something I really want you to understand. Alzheimer's isn't simply a disease where someone's brain becomes filled with plaques and tangles, and then they just start forgetting things. There's much more happening. Communication between neurons begins to break down, synapses are lost. Remember, those are the connection points that allow neurons to communicate with one another. Inflammatory processes become involved, cellular metabolism and energy production can become impaired, and eventually, neurons themselves begin to die. As more neurons and connections are lost, the brain's networks become increasingly unable to perform the jobs they once performed. Memory can deteriorate, language can change, judgment can decline. People may become confused about where they are, or eventually struggle to recognize people they've known and loved for decades. So when we eventually ask, could a peptide help someone with Alzheimer's, we need to ask a much more specific question. What exactly would it be helping? Could it reduce abnormal protein accumulation? Could it protect neurons? Could it reduce damaging inflammation? Could it help surviving neurons maintain their connections? Could it encourage new synapses to form? Could it improve cognition without actually changing the underlying disease? Or could it genuinely slow the progression of Alzheimer's itself? Those are all very different accomplishments, and we're going to keep separating them. Now let's look at Parkinson's disease. Most of us associate Parkinson's with movement, tremors, rigidity, slowed movement, difficulty with balance, and there's a biological reason for that. In Parkinson's disease, neurons that produce a neurotransmitter called dopamine progressively become dysfunctional and die. A particularly important group of those neurons lives in a part of the brain called the substantia nigra. Dopamine helps the brain regulate movement, so as these dopamine-producing neurons disappear, the brain progressively loses some of its ability to control movement normally. But Parkinson's is much more than a movement disorder. People can also experience changes in sleep, mood, smell, autonomic function, attention, thinking, and eventually, in some people, dementia. Another major piece of Parkinson's biology involves a protein called alpha-synuclein. In Parkinson's and several related diseases, alpha-synuclein can misfold and accumulate abnormally. Those accumulations are associated with structures called Lewy bodies. Researchers are also investigating the roles of mitochondrial dysfunction, oxidative stress, neuroinflammation, and impaired cellular waste clearing systems in Parkinson's disease. So again, if someone says this peptide could help Parkinson's, my next question is, of course, how? Does it help dopamine signaling and temporarily improve symptoms? Does it protect dopamine-producing neurons from dying? Does it affect alpha-synuclein? Does it improve mitochondrial function? Does it reduce neuroinflammation? Or has it actually been shown to slow progression of the disease in humans? Again, those are not interchangeable claims. And mentioning Lewy bodies brings us to another condition that's probably a little less familiar to you, and that's called Lewy body dementia. This one can get confusing because there is biological overlap between Lewy body dementia and Parkinson's disease. Remember that protein we just discussed, alpha-synuclein? Abnormal accumulations of alpha-synuclein inside neurons form Lewy bodies. And Lewy body regulated pathology occurs in both Parkinson's disease as well as Lewy body dementia. But the way these diseases present clinically can be different. Someone with Lewy body dementia may experience progressive problems with thinking and attention, changes in movement that can resemble Parkinson's, sleep disturbances, and sometimes very vivid visual hallucinations. One particularly interesting feature is that cognition and alertness can fluctuate. Some may appear relatively sharp at one point and significantly more confused or inattentive at another. And this gives us another example of why saying a peptide for dementia doesn't tell us nearly enough, because Alzheimer's disease and Lewy body dementia can both cause dementia, but the underlying biology isn't identical. If the disease process is different, the biological target may need to be different too. And then we have vascular dementia, and this tells an entirely different story than what we just talked about, because now the primary problem begins with the brain's blood supply. Your brain is extraordinarily metabolically demanding. It needs a continuous supply of oxygen and nutrients delivered throughout the bloodstream. When blood vessels become damaged, when someone experiences a stroke, or when tiny vessels deep within the brain become diseased over time, brain tissue can become injured. Sometimes that injury happens dramatically with a major stroke. Other times, damage accumulates much more quietly through repeated small vascular injuries. And eventually, enough of the brain's networks can become disrupted that cognition begins to suffer as well. And depending on where that vascular damage occurs, someone may develop problems with processing speed, attention, planning, organization, decision-making, memory, walking, or other neurological functions. And here's another complication. People don't always have only one form of dementia. Someone can have Alzheimer's pathology and significant vascular disease at the same time. That's sometimes referred to as mixed dementia. So even with one person's brain, there may be several biological problems occurring simultaneously. And now I hope you can see why the question, do brain peptides prevent dementia, is almost the wrong question. My first response would be, which dementia? And then, what part of that disease are we trying to change? Are we trying to protect the neuron from dying, reduce neuroinflammation, improve blood flow, support mitochondrial function, prevent abnormal proteins from accumulating or spreading, protect the synapses that already exist, encourage surviving neurons to build new connections, improve neurotransmitter signaling, help the brain reorganize after an injury, improve someone's memory or attention, even if we're not changing the underlying disease? Or the biggest question of all, are we actually slowing or changing the course of the disease itself? Because those are completely different therapeutic accomplishments. And this distinction is going to become incredibly important in our next couple of episodes. A peptide might increase BDNF without preventing Alzheimer's. A compound might promote synaptogenesis in an animal without rebuilding the human brain. Something might improve memory on a cognitive test without stopping neurons from dying. And a therapy might make someone function better without actually changing the disease that's causing the problem. None of those findings are meaningless. In fact, some could ultimately prove incredibly valuable, but we need to describe them for what they actually demonstrate, rather than quietly allowing one promising result to become a much bigger claim. Because when we're talking about diseases as devastating as Alzheimer's, Parkinson's, and dementia, hope matters, but so does precision. And now that we understand both the systems involved in brain repair and some of the very different problems we're trying to solve, we're finally ready to start asking, can any of these peptides meaningfully influence these diseases? At this point, we haven't talked about a single peptide treatment in depth today, and that was intentional because before somebody tells you that a peptide increases BDNF or promotes synaptogenesis, protects neurons, or helps prevent dementia, I want you to understand what those claims actually mean, and now you do. You know that synaptogenesis means forming new connections between neurons. You also know that BDNF and NGF are neurotrophic factors involved in supporting neuronal survival, adaptation, and plasticity. You also know that GABAergic signaling involves one of the brain's primary braking systems. And you also understand why HGF c-Met signaling can simultaneously make a compound incredibly interesting for neuronal repair and also raise questions that deserve scrutiny when we think about cellular growth in cancer. But perhaps the most important thing we've established today is that brain healing isn't one biological process. Protecting neurons, rebuilding connections, recovering after a stroke, improving anxiety, enhancing cognition, and slowing Alzheimer's disease are not interchangeable accomplishments. So in our next episode, we're finally going to start putting the peptides themselves under the microscope. We'll begin with Cmax and C-Link, two peptides that are frequently grouped together in the biohacking world, but whose scientific stories begin in very different places. One takes us into stroke, neuroprotection, BDNF, and brain recovery. The other takes us into anxiety, GABA signaling, stress, and cognition. And we'll ask the question that matters for both. Are these compounds actually helping the brain heal, or are some of the claims running out there well ahead of what the evidence can actually prove? That's where we'll pick up next week, so be sure to tune in. Okay, that's it for today's episode. Thank you so much for listening to Serenity and Fire. If today's episode helped you look at brain health differently and gave you a better understanding of what it might actually mean to protect, adapt, and repair the brain, the best way to support the show is to follow, leave a review, and share this episode with those you love most. Together, we can keep asking better questions about not only how we live longer, but how we protect the minds that allow us to fully experience those years. Until next time, nurture the mind you want to carry with you, protect the connections that shape who you are, never take your cognitive health for granted, And as always, keep balancing serenity with fire. I'm Krista Guigeni, and I'll talk with you more in our next episode. At Panacea Luxury Spa Boutique, we don't just offer traditional spa treatments. We create rituals that relax your mind, restore your health, and rejuvenate your spirit. From biohacking technologies to advanced oncology-trained care, everything we do is designed to help you heal on the deepest level with clean, holistic therapies, products, and amenities that are second to none. And right now, you can experience two of my favorites, our whole body LED light bed or hyperbaric oxygen therapies. And as a thank you for listening, you'll get 10% off your first session when you use the code Serenity10 at booking. What is your panacea? Let us help you find it, because true wellness isn't a quick fix, it's a ritual.